Alrighty, so again, thank you all so much for having us. My name is Nuzhat, and I am a second-year internal medicine resident here at UC San Diego. And so Shira, Annika, and I have been working together to better understand fungal resistance patterns here at UC San Diego Health and drivers for this resistance. And so we're particularly interested in the role of exposure to agricultural fungicides. So as a broad spectrum overview, fungal infections are primarily a burden for people who are immunocompromised. However, we've seen over time that this is changing. And then annually in the US, fungal infections result in over 7,000 deaths a year and over 7 billion in direct medical costs. This is because invasive fungal infections are often very challenging to treat for a number of reasons, including rapid adaptation of fungi, as well as the limited arsenal of antifungal drugs that are available for use in clinical treatments. And so while there are roughly 4 classes of systemically acting antifungals that are used in medical treatment, we'll focus in on azoles, which is a backbone for the treatment of these invasive infections. Fungal infections don't just affect humans, they're also found in plants as well. And what we're seeing is that fungicide use is increasing, both in humans for clinical treatments and in agriculture as well. From the medical standpoint, the number of humans susceptible to fungal infections has rapidly increased, and this is in part due to advancing age, medical interventions that focus on patients with cancer, organ transplants, HIV infections, generally suppression of the immune systems. And this has led to an increased use of antifungals in clinical practice. And then from the agricultural standpoint, as human population has grown and urbanization has occurred, there's been an increased demand for food and the quantity of food as well. And so this pressurizes the agricultural industry to focus in on more intensive practices like monocrops, fertilizers, pesticide use, which inadvertently selects against the defense mechanisms that plants and ecosystems have. And so this ends up resulting in increased plant fungal infections and yielding great losses in crops. So of the approximately 1.5 million fungal species that we have on the planet, there are very few that are common pathogens for humans. And this is in part due to immune factors and likely bodily temperature as well, as most fungi tend to thrive in 12 to 30 degrees Celsius temperatures, and human body temperature tends to be a little bit higher. But however, as we have climate change and the fungi evolve into a warmer climate, they develop adaptations that allow for emergence of new pathogens and increased resistance to antifungals. And some of these different mechanisms are listed here, whether that's cell wall thickening, nutrient utilization, enhanced detox processes. And so as heat increases, as climate change occurs, farmers are driven to use more fungicides to these fungi that are adapting, which spirals pressures for resistance. And one common class of fungicides that is used and relevant to both medicine and agriculture is azoles. And so we've seen a great uptick in azole use from between the 1992 to 2005 timeframe, in which that use was very constant or stable. But compared to 2016, we've seen greater than a 4-fold increase as well. And all that to say, antifungal resistance is a global concern. It's been reported in all across the world, including the US, Europe, Middle East, Asia, all throughout the different continents. And as an example, we can take a look at Aspergillus species, which is a type of fungi that affects both plants and humans. There's been an increase in incidence of the azole-resistant Aspergillus in agricultural fields in some studies. And then other in vitro studies have demonstrated that Aspergillus exposed to agricultural azoles can develop a cross-resistance to medical azoles. Along with all of this, there have been case reports of patients who have never been exposed to medical azoles before that develop infections with medical azole-resistant Aspergillus species. And together, all of these tend to suggest that there is a selection that's occurring for resistant fungi in the environment due to antifungal use, and that our patients are then acquiring these resistant organisms that result in clinical disease. And then in spite of all of this, we don't routinely test for fungal isolates from our patient samples for their susceptibility to the common antifungals that we use. Instead, our selection of antifungal drugs is almost algorithmic. And so we've discussed previously, as our climate changes, these fungal infections pose a growing risk to human health. And so our project is a little bit more descriptive in nature, and we're hoping to get a sense for the trends that are occurring here at UCSD Health and within the San Diego and Imperial County areas. So basically looking at trends in fungal susceptibilities to azole antifungals in the UCSD patient population, as well as trying to describe the agricultural azole use trends in our county and in Imperial County. And so just to look at the methods for the resistance analysis, we obtained reports of antifungal susceptibility testing that was done for our patients from 2016 to May of 2023. Most of the antifungal susceptibility testing is not done in-house. The only species we do in-house is Candida, which is very prevalent. Everything else we send out to Arab laboratories. And once we got this list, we did a manual chart review, pulling all of the susceptibility data for the fungal isolates that were tested, and then did some analysis to try to look for trends. And then in looking at the agricultural azole use data, we obtained this from the California Department of Pesticide Regulation, searching specifically for agricultural use of chemicals with azole in the name in San Diego and Imperial counties from 2016 to 2021, 2021 being the most recent data that they had available. And so just looking at our susceptibility analysis, between 2016 and May of 2023, we had 214 samples that were sent out for testing. And of those, we looked at 175 samples, excluding the isolates that didn't have any susceptibility data and patients who lived out of state. And then we also excluded Candida because the send-out data does not represent the full spectrum of testing available for Candida, since we had shifted to in-house testing, and this will need its own analysis in the future. And so here's a distribution of the patients with fungal infections that were sent out for susceptibility testing within the timeframe that we discussed previously. And so the zip code with the most patients with fungal infections that had been sent out for testing was 92114 with 9 isolates. And this was the area that's like north of Chula Vista and east of National City. Next up was 92101, which had 8 isolates, encompassing the area that has the airport, East Village, downtown San Diego, and Bankers Hill. And then third runner-up was El Centro with 7. If we accounted for everybody who has had a fungal infection and not just those who had their sample sent out for susceptibility testing, I imagine this map would look very different. And so this is a list of the top 10 fungi that were observed in our clinical infections data set. The greatest number of infections were with Fusarium. And then you can see the list has various species of Aspergillus on this as well. And interesting to note, both Fusarium and Aspergillus can cause infections in both plants and in humans. Just a brief overview of how we test for susceptibility to a drug. We look at minimum inhibitory concentration, or MIC for short, which is the lowest possible concentration of an antimicrobial drug that will inhibit the visible growth of the organism. And so higher MICs mean more of the drug is needed to inhibit the growth of the organism, which can indicate higher resistance because lower concentrations of the drug are no longer affecting the organism. And so we looked at the sensitivities for Fusarium and Aspergillus fumigatus, since these had 20 or more samples available. And the medical azoles that we looked at were itraconazole, posaconazole, voriconazole, and isobuconazole. And to summarize what we saw, Fusarium demonstrated very minimal change over time in its very high MICs. It is known to be highly resistant to azoles. And that was demonstrated in what we saw in our data as well. For Aspergillus fumigatus, we saw very minimal change in the MICs for itraconazole and posaconazole. However, we saw perhaps the slightest increase in the MICs over the years for voriconazole and isobuconazole. I'll show you what we saw for Aspergillus and the isobuconazole data on the next slide. So what you see here is a table demonstrating the MICs in micrograms per milliliter for the isolates of Aspergillus fumigatus that's broken down by year. And so lower concentrations are on the left and higher concentrations are on the right. And so if we for example, if we look at 2018, the row for 2018, there were 2 isolates tested that year, and one of them had an MIC of 1 and the other had an MIC of 2. And so if you look at the samples tested over time, you might imagine that you are seeing a rightward shift in the concentration of the drug needed to stop growth. But it's really hard to draw conclusions from this because the biggest thing that we see here is that there were very few isolates tested of this fungus tested per year. And so moving on to the data from the California Department of Pesticide Regulation, this graph compares the agricultural azole use between San Diego and Imperial counties. X-axis is the year, and then Y-axis is pounds of azole used, and then it is further color-coded by the type of azole that was used. And so you can see that the most common azoles that were used in Imperial County for agricultural purposes were propiconazole and tebuconazole, whereas in San Diego County, difenoconazole and perhaps propiconazole were most commonly used. And then this graph just compares the total azole use over time by county. So again, x-axis is time and y-axis is pounds of azole used. And as expected, Imperial County demonstrates an 8 to 10-fold higher use of these agricultural azoles compared to San Diego County, as the agricultural industry there is much more robust. And then this is just a series of maps that highlight the distribution of the more commonly used agricultural azoles in Imperial County and in San Diego County. So you have tebuconazole and propiconazole on the left-hand side and difenoconazole on the right-hand side. These maps were obtained through Tracking California, which is a publicly available pesticide mapping tool. And the data is from 2018, which is the most recent data they had available. So in looking back through our study, the limitations for this is very extensive, but we wanted to have an initial look at the data for our health system and the exposures that the people in San Diego County and in Imperial County might have. The biggest thing is that we have a very low sample size for our susceptibility data. There were only 175 non-Candida fungal isolates that were sent to Europe for testing across the UCSD health system from between 2016 to 2023, whereas their use of antifungal drugs in patients is significantly higher. There's limitations to the zip code data that we were able to obtain versus, for example, the EMR only reports the last known zip code for the patient, and for those who are experiencing homelessness, the address defaults to the UCSD Hillcrest address frequently. And so really, it's hard to draw causal links between the agricultural azole use data and the medical azole susceptibility data, but we were hoping to describe some trends within the areas. And so the other thing that we need to keep in mind is that this is in large an overview that looks at environmental impacts, such as aerosolized exposures, maybe contact and via soil, but doesn't necessarily account for ingestion of crops that are produced in these locations, because they get shipped out to all throughout the country. But the biggest strength that we see for this study is that it shows us this big gap in our knowledge and our monitoring of fungi. And so this is important because fungi are evolving and developing resistance as they adapt to climate change. And though we can't draw conclusions, we may be seeing some shifts in the susceptibility of Aspergillus fumigatus to some of the common medical azoles that we use. And so next steps for us include evaluating resistance trends for azoles in Canada. And so we have a larger set available there. Maybe we'll be able to see some trends. And then we also hope to collaborate with other UCs to grow the data set for these other fungal species that we discussed previously, which may lend some support to some of the patterns that we see. It would in particularly be interesting to see what data institutions like UC Davis and UCSF Fresno might be able to offer as they serve areas with higher focus in agriculture. And of course, biggest thing is that we will need to increase our own institutions' antifungal susceptibility testing for surveillance purposes so that we are better equipped to tackle these questions of antifungal resistance in the future. These are my references. Happy to take any questions.
Very interesting presentation, fantastic. Yeah, this brings, I think, a lot of the thought processes that we tend to forget actually when looking at a lot of different environmental determinants of like the upstream or downstream effects of a certain exposure in this case. Very interesting. So let's open it up for questions.
I had a comment and a question. Excellent presentation, very important study. One question, have you worked with people at the El Centro Regional Medical Center?
We have not. All of the data that we have is for patients who got their testing or who are patients of UC San Diego Health. But there were people who lived out in El Centro, had El Centro listed as their home address.
Okay, well, the medical center is actually connected with UCSD. And if you'd like, I can put you in touch with a colleague and a friend of mine, Christian Tomaszewski. And Chris runs the emergency medicine clinic, and he's very open to collaborations with people. So one question I had, I mean, as you know, I'm sure, I mean, Imperial County, especially the Imperial Valley, has had spikes in COVID infection. And is that something that you would take into account in terms of immunocompromised study participants? Does that interact some way with fungi?
I think that's a great question, a great point. I think most of our data will predate COVID, but there have been observations that COVID predisposes people to invasive fungal infections. It gets very complicated, so it all falls into just how very limited this initial look at the data was, but it was just to kind of start tackling this issue. But I think there's going to be a lot of different factors. It's just this factor of environmental exposure hadn't been explored. But I think your point, COVID does predispose people to fungal infections. Not frequently, it tends to be the sickest of the sick who need maximal life support. But it's a good question.
Well, one thing I'll just close out. I mean, I think you've done an excellent job with this pilot study. And if you want to pursue an NIH grant, I mean, having demonstrated that it's feasible to do a study with participants in the population, and you have some data to present, I mean, that's a key feature.
Yeah, and this was clinical samples, so it was just retrospectively looking back at those who not only had a fungal culture, because we'll get the results of the fungal culture, but then a lot of times it just sits there and that's the end. It's then sending it for these susceptibilities, which is where the gap in knowledge is. So we're growing a bunch of aspergillus, but we just presume, okay, we're going to treat it with this. Or, sometimes in some scenarios you don't treat. I'd say the majority you don't treat.
Yeah.
It's that sending off for these susceptibility data that we're not doing. Of course, there's flaws in interpreting susceptibility, and that's outside of this scope. But I think it's important, and we think it's important. But we didn't enroll patients. We were able to. This was just retrospective chart evaluation using MRNs, but because I do antimicrobial stewardship, and Annika Klein is the head of the microbiology lab. It's kind of within our scope of what we should be monitoring. And so it was kind of a quality evaluation of like, well, how much are we testing? What are we finding? What are we looking at? And then, the environmental aspect of looking at pesticides in the environment didn't require an IRB evaluation. But all that's just to clarify. Thank you very much for the feedback. And we wanted to thank you in particular, because when we were kind of brainstorming on this, you led us to the pesticide databases, which we had no idea about. So thank you, Harvey, for that contribution.
Sure, absolutely.
Yeah, those are great, amazing resources.
And just as a little extra background for those of you who are not in the pesticide world, but fungicides currently it's estimated that about 70 to 80% of all crops in the US are treated with a fungicide. And a big part of that is because they found that that can result in 7 to 12% increase in the yields, which is very substantial increase, and that was really quite an economic incentive for many crops and the plantations to be doing. And so there are a lot of different fungicides, of course, and I think going down the azoles path is a very interesting one, but there's many other fungicides that, of course, are also being used in agriculture. So something that was popping into mind while you were describing, I think this is very thought-provoking. And there's different ways that you can think of how you can further a study like this. And it could be something like an ecologic study in which you include you gather information for different health systems across the country in some places that have high and low use of antifungals or fungicides in this case, and seeing if there are differences there. And then that just gives you like a general kind of an overview, which is kind of similar to what you're doing right now, right? So you have all the sensitivities there for aspergillus, and next you're going to be looking at Canada, and hopefully you'll have a lot more for Canada, right? And then looking at within the state or within Southern California, but it could open up the opportunity to start looking elsewhere. And this is something that I have not seen anybody present on in any of these conferences that I've been attending with regards to this particular topic. And I think it's spot on. I mean, we've been within our studies in Ecuador, we have been measuring antifungals, and we just had to chop the aim we had to cut it out for the measurements of azoles in our study because of NIH funding. That's a whole other battle that we've been having with them. But the main concern from our end was not looking at resistance, but maybe we should be looking at that. But our main concern was mainly hepatic toxicity, because a lot of azoles are hepatotoxins, so we were going down that pathway. But I think that, this is actually giving me some thoughts. Maybe we should start collecting some swabs of different places. Maybe we can talk about that, and then just looking at what some of the biomarkers of exposure, how they may be related to different resistance for different pathogens. Yeah. Any other comments, questions?
I just have a question in terms of the data. I have no sense of, I guess, do you have a sense of how much resistance sort of varies person to person? Because if you're looking at, this sort of over years, but you only have a few people, of course, it's something you guys were saying, but I guess I just am curious, do you have any guess of like how many patients you would need, or how many samples you would need to really be able to make some sort of inference here in terms of the variability?
That's a great question, and we totally just did convenience and we have the data. But yeah, so what Nuzhat had mentioned was this is totally limited, because an aspect of when you start seeing resistance may be in somebody who's getting directly treated with azoles
Right.
over time, and that was out of the scope of this project. Nuzhat had 4 weeks during her residency to do this work, and we'll have her for 2 weeks to look at the Candida. But it would be, putting together all these puzzle pieces, we could go deep, and that would be going deep. We were just kind of curious because there are these reports, and even with other fungi, like there's this Candida auris that's now sweeping the world as a kind of climate change pathogen from the combination of a warming climate and increased pesticide use. Now, Candida auris is kind of this new fungal pandemic, if you will. But I guess maybe I lost my train of thought. But if we went in a deep dive, we would have to know what that each patient was exposed to, what were their environmental exposures, and so this was just a very broad overview. Yeah.
We did what we could without an IRB to start. So
Yeah.
there is already support in the clinical center to look at isolates and just describe them over time. We do that for bacteria so that we can say, if you grow this bacteria at UCSD, this % are resistant to this drug. That's called an antibiogram. So Shira and I kind of came up with this idea, really Shira did, and recruited Nuzhat. Excellence, by the way, that was a beautiful talk. Just to sort of say, well, we spend a bunch of money sending out mold susceptibility testing. It takes a while and it's not cheap. So the lab doesn't just do it routinely. It has to be requested usually by an infectious disease provider. And then it takes a few weeks to come back. So you kind of have to have a patient that's, usually it's going to be a patient that's like failing therapy or getting worse. But we don't have that data to look back at the patient charts and start collecting data about what drugs they were on and what their diagnoses are, then you need IRB approval to do a retrospective analysis. Taking the isolate and just getting a zip code for where the person came from, that was deemed to be sort of okay with our current sort of clinical care QA stuff. That's part of the reason we
Like
don't.
environmental justice.
Yeah, we don't have we'd have to, the next step is to say, okay, well, now with an IRB, we can get more information if they'll approve us to retrospectively ask a few questions.
I do think it was interesting though, Nuzhat, that El Centro did pop up as one of the highest frequencies. They're not our most frequent patient, but again, another limitation.
It's interesting. Yeah, it makes me wonder if those patients are getting sicker, for some reason. And that's why they're electing to send out susceptibility testing. To answer your question, Alexandra, about whether it's the same patients or not, running the lab, I can tell you that we don't want to spend hundreds of thousands of dollars on isolate testing to a send out lab. So most of the time patients are only going to get an isolate tested one time. So they're not going to have multiple data points over time for resistance testing. Whether they grow it over and over is a different question, but these were only the isolates sent out for testing, not all of the isolates. Certainly patients will continue to grow aspergillus for many years. I mean, our cystic fibrosis patients do that. They never clear it. So at least her data set's probably individual patients for each mold, or close to individual. Sometimes maybe someone grew 2 different molds at once, but yeah.
Yeah, very, very, very nice. I mean, phenomenal work, phenomenal presentation indeed. And we'll be switching over now to Alex, and while she gets ready for that, I'll sneak in one last question here for you. I'm looking at the National Database of Antibiotic Resistance Organism. Are you familiar with that? And do you know if you can get a zip code or some sort of geographic unit linked to specific or prevalences of antibiotic resistance if you want to look at different species?
Are you talking about NARMS? The NARMS Now platform?
NDARO was what this one is, NDARO. And then there's NARMS. Is that what you're? Yeah, either one. Are you familiar with either? Is there any data that could be extracted that's also linked to a geospatial, some sort of geospatial construct?
They definitely have something geographic because you can click on individual states, I think, for organisms, I believe. Usually if you pull those databases and you download the CSV file, they often have a lot more data than what's presented in their graphics interface. So I haven't personally tried to do that, but it wouldn't surprise me if you download a CSV file from one of NARMS, if you can get more line item information about where the isolate came from and what zip code it came from, or what public health lab it came from, which would tell you a lot. So
yeah, I mean, you could think of as I said, an ecologic study in which, if you have those data, then we can start plugging in different zip codes that we know high or low pesticide use, and then comparing that across the nation if it's easily available. So I think there's a lot of questions here. I think we have many more. So if you didn't get a chance to ask the question, please post it on the chat. And I see that Jenny has also included some questions. So please also feel free to answer to some of the questions since we don't have a ton of time here. But thank you very much.